guys hear me? Hi, I'm Sarah Bowen. I am from the Barrow Neurological Institute in Phoenix, Arizona. I run a biobank, a tissue repository, and several other things that are fun. I have you noticed that you can put post-it notes on the board that have ideas of things you want to do next year? I highly recommend that you do that because I put a post-it note that just said, "Are there organoid cores?" on a post-it note last year. And I had the brilliant idea of adding my name to that post-it note. And so here I am. Please add your name. Because I added my name, I got to talk to these two very lovely scientists, and I'm learning a lot just in the conversation we had before the meeting. So I have a few housekeeping things to say. You are in the Emergent Technologies meeting. Please silence your phone. Photography of ABRF meeting sessions is permitted subject to speakers' requests not to capture pre-publication information. Recordings of the program sessions will be available on the meeting website following the annual meeting. And I have permission from both speakers that we can share their slides and their session can be recorded. ABRF social media policy is that everything can be shared. If a speaker asks that it not be shared, please respect the speaker and do not post on any social media. Hashtag for the event is hashtag ABRF 2026. Unless noted by the speaker, questions are to be held until the end of the session. Attendees may ask questions at the microphones in the room. Obviously, we're recording this, so we would like to have your question recorded too. Download the ABRF app to review the full schedule, access lightning talk abstracts and posters, connect with other attendees and sponsors, play the ABRF game, which I am dominating at currently, and create your personalized meeting schedule. You'll find extra content, including hours of exhibit hall and tech showcases, along with resources from speakers and sponsors. All attendees will receive an overall meeting evaluation survey following the meeting to help improve the ABRF meeting experience. You can also provide feedback for each session in the app. Look for Evaluate this session link within the session. Like I said, I was really interested in finding out if there were organoid cores, and apparently there is one in this city. As we're shifting from animal models possibly into more human-based technologies for preclinical research, I think that these two technologies are really going to help move us forward in that endeavor. Polly Mattila is from the University of Pittsburgh. She's a director of Organoid Research Core at the University of Pittsburgh. She's done a lot of research as a cell and molecular biologist, and now she's learning about the core experience, building this organoid core from scratch. She has a BS and a PhD from the University of Minnesota, and it's her first time attending the ABRF meeting, so if y'all can make her feel really welcome, that would help.
Thank you. Let's see here. All right. So like Sarah said, I'm Polly Mattila, and I have two different titles. I started at the University of Pittsburgh in the Institute for Precision Medicine. So our focus is on a lot of preclinical advancements, personalizing medicine. And I was tasked when I entered, or a few months after joining, I was tasked with looking into how to do a little bit of cost recovery with some of the organoids we had. And that turned into creating a core. So today I want to share with you a little bit about organoids, creating a core, and like Sarah said, the motivation or the movement towards using human models. And then I want to share a couple successes. Okay, so this was a image I thought was really cool. It looks really futuristic. It looks like the possibilities are just unending. You've got pharma, you got pharma in here, you've got scientists going on, we've got a brain in this thing, and all these things connected. So there's a lot of, if you start looking in the literature, you see there's just a ton of different types of organoids and related technologies with that. So this is just to give you a little sampling of all the different things that keep popping up. And if I'm constantly, for the Institute, I'm always looking at the new emerging technologies as well as new medicines, new clinical trials. So in this one, I thought this was actually kind of pretty, being a cell biologist, seeing this, the stomach organoids secreting insulin, tests in paralysis, repurposing of drugs, chemotherapeutic toxicity. This one is psychiatry. And this was a recent editorial, and the title was really catchy to me. It was From Models to Machines, Why Organoids Are Becoming Infrastructure, Not Just Insight. And on top of that, you can see that the predicted market for organoids is almost $12 billion by the year 2035. So there is a lot of momentum behind this. So what I'm going to do first is kind of set the stage here and talk about what our traditional models have been and what we're really comparing organoids to. The talk after me will be talking more about the chips, which is another technology, our human models. In this case, animal models have been really crucial in drug development, but they also face significant limitations. So one of the things we all know is there's species variations, and one of you can see that only 10% of the drugs that go into trials actually make it. So that's a pretty high failure rate. Furthermore, a lot of them can fail from the toxic effects in humans. So it may work in an animal model or screened in a cell model, and as soon as it gets into the human clinical trials, what they find is it's too toxic and they have to end it. So this has spurred an urgent need for models that are more relevant to human physiology and represent the human response to drugs, to therapies more closely. So I'm going to talk about the organoids here, and sort of using the animal models and cell culture as sort of where we're currently are more traditional models. So briefly, organoids, if you don't know what they are these 3D structures, and they are taken either from iPSC cells or pluripotent stem cells or adult stem cells. They are put into extracellular matrix, and probably most people have heard of Matrigel or Cultrex or something like that. There's this big industry now looking at synthetic ones. And what that allows are for the cells to form these structures. The cells then can interact with each other and interact with the extracellular matrix, which is more closely mimics what the human physiology would be in an organ. So you can imagine on a flat plate when you're interacting with plastic and you're spread out, even the depth of it and the way a cell would signal is very different than if you had your neighbor cell next to you. And I'll talk a bit more about what we do, the adult stem cells from tumors. These cells are heterogeneous, so you get this mix in there. And it's the autocrine, the paracrine, the interactions with the extracellular. So you get this more complex interaction and structural structural model than you would in a 2D. So as I mentioned, there are a couple of different ways you can do them, from the pluripotent stem cells or from adult stem cells. And this is what our core does. One of the reasons for this is they're very different, and the expertise to do both of them are very different. And I'll kind of get into that, since we are a very new core, we wanted to limit we wanted to focus on what we did well. So organoids are also way more cost effective and a higher throughput compared to animal models. And as Sarah mentioned, there is, if you've heard, or probably most of you have, the NIH has now prioritized human models in what they call these new approach methodologies, or maybe you've heard the term NAMs. And they are really advocating for people to build these models that have a higher human relevance, and this can help replace or reduce the use of animal models. So these are just some examples of where organoids have been or are being used in. You can see that they are used in disease modeling, drug testing, studying development, and regeneration of tissues. They have some advantages. Like I mentioned, they have a better prediction for drug efficacy or other therapies, as well as an earlier detection of toxic effects. And both of these really contribute to a cost and time saving. So you can imagine if you are doing preclinical screening, you can do a lot more drugs or therapies, and you can find out sooner which ones may be a better choice to move with. And the other thing, like I mentioned, being part of the Institute of Precision Medicine, we really are focused on individualized treatment strategies. So an example of that would be knowing the genomic profile of a cancer patient and being able to screen different therapies and seeing how those affected that person, because each person may respond differently to a given therapy, and that can help guide clinical decision. Now, they're not perfect, and they do have limitations. Right now, one of the problems is the standardization of protocols. How are you developing them? How is that different from the other lab? How do you compare those results? And I think that's just something very common in research anyways, right? Can we repeat what we're seeing? If we're going to call it a preclinical model, we need to have standard protocols, materials, and how we're reading that. The other thing that is somewhat limiting, although I would say there's a lot of research going on right now, and there are models being developed with vascularization as well as immune components. All right, so this is just to give you a feel for the timeline of all this priorities. It got a little overwhelming, and I've heard it at our university where people are somewhat, especially if you work in an animal model, they're like, I have to get rid of it. I need to adopt this. And they may not even have the expertise to do cell culture. So you don't have to get rid of it right now. It is a process, and what our core does now is we try to go out, outreach, let people know what we're doing and how we can help them, and let them see how these models might fit into their current research plans. So the NIH recognizes that there's a rising demand for more accurate, human-relevant disease models. And they started back in, and there I went and pushed the wrong button. Okay. Back here, they started with a working group, just kind of surveying what people needed, what they thought they could do, and made some recommendations to NIH. And that they accepted those recommendations. And this is what the NAMs are. There's a number of things. They can be AI, organ on a chip. They can be tissues. They're just things that are relevant and can be used in combination. And you're seeing a lot more of that, a lot of AI now too, that is being incorporated into the studies. Then there was this announcement with the FDA. And their statement was that they want to make animals an exception, and the FDA requirements can shift to a NAM-based default. And just within the last couple of weeks here, they released guidance, a draft on the guidance to alternatives to animal testing. So this is moving along, and there's a lot more stuff out there. Just in March, there was a whole bunch of announcements. The NIH is also supporting this movement. They're offering, they're funding it. This is a statement that they just had, I believe this was the third week of March. This came out saying that they had $150 million that they've invested in these human-based models. The FNIH is a foundation for the National Institutes of Health, and they've already had one round of submissions, and they chose some, I think it was like 4 or 6 different groups to work with these. Now they are partnering also with industry and pharma so that they can work and move towards validating these models. And then NASA has a fun tournament that they've done, I believe, once or twice now, and that one they just finished up. So they are trying to reach out and support the research community by funding it, not just saying that it needs to be done. And what I was interested in here is this standardized organoid modeling center that was announced in September of 2025. And this is really an initiative to, like I said, standardize the protocols, the materials, make organoids in a way, and then make those protocols as well as the organoids available to the research public, the researchers. And let's see here. All right. And these are just some examples of real world use of organoids and organoid-related technologies. So you can see there's a number of companies, and it's anywhere from pharma doing it to just industry making organoids for people, contract research organizations that screen and use organoids. And this one was right here. This human on a chip replicates neuromuscular disorders, finds a new treatment without animal testing. So there are trials already going on and being approved. All right, so this is the big question. So when this started, and we are very early on, our core will be 2 years in July. So when we first started, we really were we had this large biobank, and researchers were asking for these organoids, and we were like, wow, this is getting costly to distribute. So then we thought, well, maybe we'll use one of the other cores. But the university thought maybe it would be better if we started a core. So here we are. We did start one. And just to remind everyone, cores are really, really valuable. They, in my opinion, should be just sort of standard funded by the university, not just on soft money. I think it should be hard money, because I think cores offer such a benefit in a number of things by expanding access to everyone on campus. So for example, if you are a researcher, and we have a lot of physician scientists because we have a hospital here in Pittsburgh, physician scientists come in and they may not have, especially early on, may not have technicians or have the expertise, but they want to do a research project. It's nicer if you can work with a core. Also, the cores elevate voices, so we can act as as I just got done listening to the advocacy, and so I'm going to say we can advocate. We can, instead of an individual saying, I need something, we can say, the community needs this, right? Obviously, we try to reduce waste by centralizing some of the resources, cut supply costs by working with vendors if we can, and then building connections. One of the things I do all the time is I have people who, because the name of our core is the Organoid Research Core, so we pop up really easily because everyone says Organoid Core. That's the first thing that comes up. I get from all over the country people asking me, can we make this? Can we do this? Can we train with you? We're not quite at that point where we can offer training for groups, but I mean, this is what people want. This is what people need. We do it on campus right now. But I also get questions from people all over the university asking me, do you know anybody who uses neuro organoids? Or do you know anyone who uses cardiac organoids? And so I kind of sit in this centralized place where I do a lot of outreach to a lot of different groups and try to connect people and build this kind of nice web. We don't have to be the experts in everything. We can't be. So being able to know who's on campus is a really good resource for everyone. And then again, I'll put my little pitch in here for advancing precision medicine. Personalized treatments, that's what is out there right now. How can we, instead of targeting the general population who might act on that bell curve right in the center, what are the outlying? How do we personalize medicine? How do we personalize treatments for people? And then, of course, cores raise the visibility of a university. All right, I won't go through everything on here. This is just to give you a feel of like starting out. If you haven't started a core, it's a really interesting process. So when we started, oops, sorry. There we go. Okay, so when we started, this was, like I said, back in July 2023. One of the first things we wanted to know is whether or not we should start this core. So we did a survey, and we had like 4 things that we knew we did already well. And then we had a couple open-ended questions on asking our community, what do you need? And we got great feedback. So that helped us kind of like focus on what services we might offer. Then we did a business plan and a cost model. I mean, these are small businesses, and having all your ducks in a row is really important. We went to a committee, we got approved, and then, like I said, we had our efforts that we were going to do very focused. We wanted to know, one thing you want to keep in mind when you're setting this up and starting one is, what are your staff's capabilities? I mean, how many staff do you have? What are their expertise? You don't want to go outside of that and learn on the fly. You just kind of want to focus small, offer what you can, and then what resources do you have available? Do you have space? Do you have equipment? All these different things. All right. So we had to build a website. I had to train and learn how to use iLab and do the billing in there. So there's all these things that come along with doing a core that I had no idea, and I was never an administrator before, so now I feel like I know a lot about billing and Drupal, which I never thought I would learn, for websites. Then, as this is kind of our year 1 here, this is kind of the setup time that we did. The other thing we started doing was really increasing the awareness. And we did that many different ways, but we reached out to Stem Cell Technologies to kind of get a pit bulk pricing, and did some demos, invited people from all over the campus. That sort of gave us some visibility. And then I give talks. I meet with regulatory groups like the ICOOC and IRB, explain what organoids are. And then finally, this last part where we are in is this year 2, sort of looking at how we're going to be sustainable, what services we might want to add on, And then also, who are we collaborating or partnering with? All right, so I won't go through all this. This is just the basics of our core. The only thing I'll say is if you're starting anything where you need to have aseptic technique, you need a dedicated space. And so I said, I need a cell culture room. I don't want to share a cell culture room, and that's what we had initially. And it was fine, but it is one of those things, we get one shot at a patient sample, and so the last thing we need is a bunch of people in and out and doing things. So that is probably one of the most important things we had. We have lots of microscopes around the campus we can get to. Although we did help and invest into a high throughput one recently that has a encased or a incubated environment. So I mean, it's really variable, but like I said, the main thing is having that space dedicated for you. And that is always a premium at an institution. Again, for services, I would say focus on what you know. We do biobanking. Like I said, I'll talk a little bit more about the biobanking. We culture organoids for people, and we'll hand them off, or we'll train them. We do screening for different compounds. We look at toxicity, viability, the efficacy. And then right now, probably our biggest push now is to do a pan-cancer patient consent. So with adult stem cells in the cancer, we need to do patient consent. Right now, our group has a breast cancer one, but we're connected to the Hillman Cancer Center, and there are cancer researchers who want to do everything. Pancreatic, lung, brain, all of these different things. And so we are hoping to streamline that by offering that to our researchers. So just so you understand, like, this is the pipeline of how we do make adult stem cell. We are affiliated with, or we have partnered with surgeons. We have someone who consents. We have pathologists that gives us information, the cancer registry, and then we have communication just to even pick up the tissue. So it's kind of an involved front-end process. Once it gets in the lab, it's processed. It's put into whatever your choice of extracellular matrix is, and you can see this is about 3 weeks is about, on average, what we find for these breast cancer organoids take before we biobank them. And then these are our core services. We always grow them out, biobank them, and one of our quality controls is to then thaw them, grow them again to make sure they actually expand. We have a minimum number of passages that we do so that we know that they will be, we're trying to standardize, so we have a minimum number before we even do any assays on it. And then we do a lot of molecular characterization, whole exome sequencing, RNA-seq, STR profiling, biomarkers, different things like that. And then we share that with our researchers. And this is the result of our efforts in the biobank. We have over 150 now breast cancer models. And as I was talking about with Sarah, as I said, you know, you don't have to be a breast cancer researcher. We work with bioengineers, and they use them to test methods. Light sheet technology, they're like, hey, this is a perfect thing to do, right? We can look at tons of organoids in here and look at the markers on them. So we do a lot of work outside of the breast cancer as well. And then I just listed, am I going along? Am I over time? Sorry, this is longer than I thought. Anyways, so you can get the slides, but some of our quality control measures, like I said, standardizing is super important. Knowing what your reagents are, keeping track of this, characterization, and then just contamination and understanding those things. And so just to share a couple of successes, this is our academic researchers. So we've done a lot of different, these are just the published ones. We have a number of projects ongoing right now. Okay. So this is just to give you a flavor of that. So we work with a lot of different, we've done drug screening, we do cell signaling, we've done stuff where we've grown them and people have run them on the flow cytometer. So we do a lot of work with all sorts of researchers on the campus. The other new, other piece of our, one of our successes is working with industry. And this is what we're trying to do right now is work to develop diagnostic tools and new therapies for a very aggressive breast cancer, which is triple negative breast cancer. And the reason this is important is we're targeting these 30% that don't respond to the first line of therapy. And so we're working with a couple companies to look at drug development and also at building a predictive model that you could screen the organoids and predict the patient response. And that would help with the clinical decision making. And finally, this is the hub and spoke model that we represent. So we have our core services, but now we have also started working with other centers and using this as a way to provide administrative support so that each person who's doing this, if they're trying to do cost recovery, they aren't having to reinvent the wheel. They're not having to do all their administrative work. So we do their billing and we try to help them that way and support them that way. And then that way, the expertise remains in their hands. We don't have to learn how to do IPSCs, but we make it available to the community. And so, again, this is just some of the support we've provided, outreach. We've worked with over 50 labs now in the last 2 years. Derived a whole bunch of new PDOs, or patient-derived organoids. We do a lot of grant support, preliminary data, letters of support, and then this administrative support in working with industry. Okay, so this is, I kind of forgot to mention this, but Adrian Lee and Steffi Osterreich are at University of Pittsburgh. They're the ones who had the funding, the guidance, and they are the ones who had created this big biobank and really had the efforts put behind it. Daniel Brown, I want to recognize him because he is our senior scientist, and he's really done all the work on, keeping these as quality organoids. All right, so sorry, didn't mean to rush on the end there. Thank you very much. And then I'm just going to give you one more opportunity here. If you're interested in learning more about the novel approach methodologies, visit Institute of Precision Medicine on LinkedIn. We actually have Christine Happel from the NIH who will be talking about that. So this might be a good resource. It's on April 21st at 10 a.m. Eastern. So, sorry. All right. Thank you. Sorry, we ran it all.
So we're gonna hold questions to the end because we have a lot more exciting research to talk about. Dr. McGrath is coming at this problem from a little more engineering side. He is a former Sun Devil, like the most brilliant scientist in the world. Let me get out of the way so he can tell you about his technology.
Okay, I don't know which one of these pointers is going to work, so I'll just quickly say thank you, and then I'll test my pointers. So thank you to Sarah, thank you to Tim for this opportunity, and thank you all for being here. This is really an opportunity for me. I'm a professor of biomedical engineering, and I have this itch to start things that are sustainable and help others, which is not necessarily what typically goes on in an academic lab. So I've founded many companies and started several cores, and I'm chronically frustrated with the need to explain the value of these to administrators and fellow faculty. So this has been an amazing community to be among, because nobody here needs to be convinced that this is how you do exactly that. So it's been a real pleasure to be here. And thank you, Polly, for taking care of my first 5 slides. Let me see if they advance. I have a I have a little bit of some very similar things to say with a slightly different set of graphics and maybe a little bit different tape. So what Polly did say is that there's a major driver behind some of the sea change we're experiencing with the adoption non-animal models. So I'm going to use the acronym non-animal NAMS to mean non-animal models. There's another acronym on this slide, which is microphysiological systems, or MPS. This space is filled with acronyms that people need to learn in order to sort of walk through it. Many of them mean the same thing. I got a slide on that that maybe some of you will want to take a picture of. But the biggest driver, I believe, is the drug development pipeline, which Polly did talk about, in which 90% of drugs fail entering the clinical pipeline. There's a number here, $100 billion of wasted effort in those failures every single year. And then obviously this is not only raising the price of drugs, it's promoting human suffering. It's a major driver for moving on from these guys, right? Because they're getting a lot of the blame. So these animals are not always predictive, are often not predictive of the toxicities and the failed efficacy that is to come as we sort of move down this clinical pipeline. And so the alternatives, there are several alternatives. Sort of these new approach methodologies or non-animal models. The one that we are focusing on at the University of Rochester is microphysiological systems, also known as tissue on chips or organoid chips. So you could just consider all of these things synonyms and you will be perfectly fine. What happens here is that we organize, let me see if this pointer works. It doesn't, so this is a pointer, right? Right there. So what you do is you take human cells and you sort of organize them. A little bit different than the organoid, but sort of spontaneously, magically turns into something. Here we're going to deliberately place things in spots and try to organize it ourselves into a tissue chip. One of the usual advantages of these is that we add fluidics right away. So when we have fluidic interactions between compartments and also with drugs coming in and things like this, or effluent coming out. So there's really it's a distinct type of technology. So you can even arrange these in the form of assembled tissue models to create something of a body on a chip. The transformation from a microphysiological system into a drug development tool, which is one of the things that we're focusing on in our center, is really taking advantage of the fact that you can array these. You can actually array the fluidics as well, and then you can automate the data that you're extracting. So these are things you can't do with animals, right? So there's really an opportunity, if you can get these tissues right, to really start turning up the discovery and turning up the screening. But for all this, you need to have reproducible systems that are very well controlled, and it's very important to focus on a very particular context of use, because these are not really organs. And even though they're called organs on a chip, which I think is a bit of a vanity term, they can be very specific tissue function on a chip that can actually get you some kind of mechanism of action or toxicity mechanism or something. That they can do as long as you focus them. So there's not just this drug development pipeline. There's another major sort of driver, I believe, and that is society sort of falling out of love with animals as a research tool. And the evidence for that is this board of animal welfare groups that are growing in number. This is this is more than a year old, and there are many more of these now. So these just keep coming online. They're not only growing in number, they're also growing in power, and that's evidenced by the asterisks that you see here. Every one of those organizations is actually providing funding for non-animal research, and often funding at the graduate stipend level, where there are actually quite a lot of young people who are sort of similarly aligned to this philosophy. Okay, so this is another major reason this is happening. I think it needs to be addressed. So the big movement, the way this actually all gets movement, Holly did speak about, is these stakeholders at the federal level actually starting to change policy, right? And the big one is the FDA. This roadmap, and I'll go into the roadmap too, just really briefly, really does sort of light a fire under the entire system to start to adopt non-animal approaches to preclinical research. There was an announcement that came out of the FDA last year. Some of you may have seen or heard panic at your institutions, when it actually was originally said that they're going to end animal-only studies. That turned out to not be correct. And then they came out with the correct announcement, which is that they are no longer going to support or demand that you use animals in your research. This is a very big difference. Okay, so still okay to submit a grant to the NIH that only uses animals, although people are now afraid to do so, or increasingly, and they do need core facilities. I think absolutely every institution needs some way to help people on board with these things, because most research groups do not know how. Anyway, that's a bit of a tangent. The EPA is on board, and as Polly said, the NIH is actually getting involved in the terms of very large initiatives and funding initiatives. I want to not be the person who tells you that these are the end-all, be-all, that these are going to replace animals, even though this is wind at the back of people like myself that try to make these chips. They cannot do whole-body interactions. They will not predict toxicities like behavioral dysfunctions, right? They cannot tell you where the drug's going to go in the body and biodistribution. Animals do these things. And so I think it's really important that we do this responsibly and not just take advantage and leverage off of these changes, because I think what will happen is the reputation of these will get hit too. These do not solve everything, but they do solve some things, again, if we're very specific about what we ask of them. Here's the FDA roadmap. This came out only in April of last year. What really is remarkable to me is just how fast they propose to adopt this rarity of animals in preclinical data. I don't think they will achieve it. Have to be a little bit skeptical. But there's really good evidence for why it should be done, or it should be a focus inside the document. And one of the priorities is the adoption of non-animal models for monoclonal antibody drugs. And there's some really impressive case studies where monoclonal antibodies clear even non-human primates totally fine, and they go into first-in-human clinical trials with healthy young adults and nearly kill all of them, right? Very impressively bad predictions in animals, particularly when it involves the immune system. And so that's where these chips can really have an impact and are actually absolutely necessary. The FDA roadmap is here. It's 2029. That'll be here in less than 4 years, and they want animals to be rare. This is unlikely, I think, but it is nonetheless a fire that's burning, and people are trying to move towards it. Here's that graphic I told you about. This is the terminology. I'm going to try to move a little bit quickly through it, but there's a lot of things that fall under NAMs, and microphysiological systems, organoid chip, and tissue chips should all be considered the same thing. There's a little bit of definitions here that you can sort of look at later that will help you sort of parse out why these different terms exist, what they mean to different people. But they all fall under 3D cell culture, complex in vitro models, along with organoids and spheroids, right? So these are just non-typical in vitro models, typical from a traditional sense. They're complex, they're 3 dimensions, and all of this falls under that. But that's not it. Non-animal models, NAMs, also include tissue explants, computational models, and even cell-free systems that can be adopted. And the FDA and the NIH are both looking very hard at these as new strategies for discovery and for drug development. I'm going to show you what we do now, and then jump into the Rochester-specific part of the talk. And here's a look at our tissue chip platform. This is a tendon on a chip, and I picked it because I think it's the most sophisticated looking version of it. There's 5 cells that go into it. There's a tendon compartment on the back that actually becomes fibrotic over time. There are bars there that sort of help this fibrotic tissue not completely collapse into a ball. And then there's an apical compartment. This is the top part of the chip with fluid ports and a well. This is an open system as it's currently constituted. There's a special membrane technology, which is the homegrown version of all this, with micropores and nanopores. And we can plate a vascular layer on this and then piece them all together in this reversible fashion. And then there's a very complicated workflow that comes along, right? And so this is one of the things that needs to be taught and learned in a tissue-by-tissue manner. So here comes the initial tendon on the chip. We usually make that tendon component first. It matures over many, many days. And here comes the vascular compartment where we seed the endothelial cells. They grow to confluence. And then we actually add immune cells. As I mentioned, immune cells are really a big advantage of these chips and the place where they're really, really needed. These can actually crawl over chip where they will sort of drive that forward, that fibrotic process. So this is being used in laboratories at the University of Rochester to discover ways of sort of interfering with that fibrotic phenotype being developed with drugs. And it's just one example of what we're doing with these chips. This is our center. So this is officially a center at the University of Rochester, and it was funded by the NIH as one of 4 places in the country where the microphysiological system should be converted to a drug development tool. These are our missions, and we laid these out as part of the proposal to really give ourselves some benchmarks to go for, but here's how it looks graphically. So we live at the center of an ecosystem where we really want to do a lot of things, and we're mandated to work with the FDA on developing regulatory versions of these chips that the FDA can use to actually clear drugs. We also work with pharma on discovery tools for screening up front which drugs should even enter the pipeline in the first place. It turns out those are different customers. We did not know that when we began, so we had to split this focus. We are working locally through seminars and training programs and mailing lists and doing everything we can to try to help people learn about this technology and on board. For the FDA drug development, we have a consulting group that we work with that helps us guide some of the documentation we provide. We do have manufacturers that we have to bring on as partners in order to make these chips work. We have academic partnerships both inside the center that do reproducibility studies for us and outside the center that are just interested in partnering and adopting. We are fortunate to have several pharma partnerships lined up right now. And then we also are doing our best through websites and a service center that I'm going to talk to you about, actually distributing some of the technologies and knowledge to the world. But here's the hard part. This grant is not renewable. It's a 5-year grant, and we're 2 years into it. We started our 3rd year, and I need to somehow make this a sustainable resource at the end of 5 years. And so I'm starting to think small. What element of this do we currently have that I can keep going when the grant ends? And then maybe after that, I'll think about scaling it up, right? So what's a survivable nugget that exists? So here's what we do now. We have 18 active labs of 30 different projects, so many labs and multiple projects. There's many in the pipeline. 10 is not doing it justice. There's many different grants that are listing us now as one of the things that they do. There are 5,000 of these chips that we distribute every single year. We have multiple formats for multiple tissues. We have all kinds of accessories, including fluidics, and a lot of technical notes. This is an awful lot of work to sustain this and get this off the ground. We also have a quality system. This is necessary for the regulatory angle. So we have 50 SOPs. We work on multi-site reproducibility. And we engage the FDA directly, and I'll show you some of that. When I think about this, the minimum viable service unit is this resource core, where we help people get access to the chips, we give them access to protocols, and we send them on their way, for the most part, to do it on their own because we can't really, we're not a big enough operation to do it for them. This is what that looks like. So there's our website, TraceBMPS. You're welcome to look at it. Here's a protocols page where we're publishing some of our more basic protocols. We have a shop where people go and buy components that they can put together, and that works through PPMS. And they can put these things together in their own lab, and they can come back to the shop, load it up with credits, and continue to just basically support their work. And then we have these technical notes that go into a blog that we publish as frequently as I can get my students to put things up. But we need to have more of a community-based support of that resource as well. This is what our core looks like. The shocking thing is how small it is. When I add up this is what's supported by the U2C grant. We have an operations manager responsible for all the QC, a cell culture technician, and then a business manager, which I borrowed from another core to also help me with this core. When it's all said and done, we actually have about $330,000 worth of money flowing through this thing, with $144,000 of it being supplemented by the U2C McGrath grant. Okay, so 5 more minutes. So somehow we have to replace all of that. So that this is what the business model is. I need to get this number to 0 by the end of year 6. And even when I'm done, I will have less than 1 FTE in this course. So this is really humble. But the truth is, as I bring this forward to our administration, we're not getting the kind of response and interest that we're getting, that Polly's getting from hers. And so I'm doing this somewhat lonely operation, and maybe talking to all of you, I'll get some ideas of how to break through at the higher levels. So the real big deal, the way we really grow is by working on this quality system effort, really sort of trying to provide as a service, engagement of the FDA. A quality system is really what ensures reproducibility, traceability, and regulatory readiness. This is difficult to do. It's a very advanced skill set that we're developing and learning in real time. It involves putting together protocols and data management structures. Really everything ends up in protocols.io in our little world. And doing reproducibility studies like we've done now, where we actually set up these devices for various contexts and work reproducibility internally, different months, make sure we can set everything up, pick everything down, and get the same answer every single time. We're doing that again with a more complex system. You can see the protocol organization dashboard that's getting very sophisticated. Again, it's many, many protocols coming together in a large protocol. We've done, again, this internal reproducibility at different months, but now we've actually gone out and asked our IT to reproduce it. We've asked Duke to reproduce it, and they have. So we're getting better and better at sort of building a quality system that takes care of the quality of all the components, but actually demonstrates that the quality exists because we can get all the way to reproducibility. This is an example of us engaging the FDA with letters and regulatory requests to participate in their program. These are all negative responses. Don't look at the details. But we just had a slightly positive response for the first time. So here's my final little slide of the acknowledgements. So there's a big opportunity, and there's uncertain times, but it's interesting that the wind is blowing in this particular direction. It's one of the few places that's getting new funding right now, new initiatives. So we're building a service center. We're calling it Trace Services, and this is meant to be a sustainable center. And we really do need to build and grow in order for this to survive. There's a lot of challenges. These are hard questions. I was hoping people in the audience can help me answer them. How aggressive can we be? If we try growing too fast, we have such a small team, we'll crash and burn the team before we can actually grow. I don't know how to do this organically. We're not getting institutional support. The quality system approach services, I don't really know who the customer is for that. It seems like there should be customers. Maybe those customers need to be pharma. Maybe those customers need to be commercial. I really want to serve the academic community. It needs help with this new era, but maybe the money isn't there, right? These are hard questions. So let me just acknowledge the rest of the team. Jonah Damo, in particular, is in our Clinical Translational Science Institute, and she's the regulatory specialist who has allowed us to build the quality system, which has really been an important partner for me in this trace effort. I have PIs that are experts on tissue chips, both at the University of Rochester and Duke. Obviously, our funding, our manufacturing partners, and I think that I can stop. Thank you.
All right, we have this conference couldn't run without our corporate partners, so we'd like to give one of our corporate partners a moment to talk about their work. Do you have your? This is Meteor Biotech, and I actually have not interacted with them, so I'm learning as much as you are.
Hi, thank you for giving me opportunity to talk about our company and the technology that we have developed. My name is Amos Lee. I'm the CEO of Meteor Biotech. We are a university startup company with a technology that was developed in Seoul National University, South Korea. We make a spatial cell sorting instruments. It's a world first concept of sorting cells from tissue or organoid sections. Since this is an organoid core session, I will try to make it as much as relevant as this session should be. So, Meteor Biotech develops this Cosmosort instrument that commercializes or instrumentalizes the core technology called SLAX technology. It's a short for spatially resolved laser activated cell sorting technology. So it's a basically laser punching system from the tissue section or organoid section after you examine the histopathological information or staining information. And either you could do it DIY or you could use AI or image processing software to basically define the target that you want to analyze, and then you can get the DNA, RNA, or protein data out of it. So you can run NGS, mass spec, any kind of PCR machine, any kind of molecular biology instrument afterwards, so that you have a good connection of the histopathological information to the molecular information that you can get. So you can use IHC, H&E, fish staining, or you can use AI-based pathology. Or recently, our customers use next generation omics, spatial omics technologies like Xenium, or Akoya, or Lunaphore, and so on. So after you stain them, if you look at those methodologies as another way of staining, you can know where to punch out and conclusively get their DNA information, RNA information, or protein information. This could be very important in organoid research because if you have multiple organoids that was treated with different drugs, for example, or different viruses or microbiome and so on, you know where to, there is a specific region. You don't have to analyze all the section. If you have your target, you could just punch out those regions and then analyze their omics data very deeply. So this is a real-time video of sorting out the parts of the tissue. This was an H&E section of breast cancer tissue, and we can get them in 96-well-2 format, so that it's a speed of target per second, and therefore use downstream technologies like NGS or mass spec or other molecular analysis instruments or technologies. So the basic core of this technology is to examine the spatial or staining information of the sections, and then possibly punch out the cells so that you can use them for deep sequencing using NGS or mass spec. This is another video of sorting out single cell from a bone marrow smear. And we were selected as the first place in 2024 in top 10 innovation by the scientists, and we were able to publish some papers in Nature Reviews Cancer and other journal articles. So we do have different white papers in DNA sequencing, analyzing their copy number, single nucleotide variation, structure variations, and more complex structure variations like catages or chromothripsis, and RNA sequencing, deep full-length RNA sequencing data, so you can get isoforms and BCR, TCR sequences, and RNA mutations. And proteomics using low-end mass spec as well as high-end mass spec like timsTOF2 or different counterpart by different companies. So this is being used by different medical doctors and researchers around the world right now, from discovery and development to FDA post-market safety monitoring. And I'm just going to skip through this part. We have used this in drug discovery in triple-negative breast cancer tissue with the chemo resistance to paclitaxel. And we are using proteomics, LCMS, to identify different protein targets for discovering ADC targets. And also metabolomics is possible if you sort different parts of the region and analyze their amino acids, organic acids, and sugars. And finally, I want to show you the research that we did with organoid. This was a case where we infected Zika virus to brain organoids, and we were able to see different sequencing data from the virus itself and the host. So you can analyze their gene expressions, their mutations, and trajectory, how these viruses spread throughout the organoid, and how the host is reacting to the trajectory of the virus. So that's the thing that I wanted to present to you today. And we do have our customers currently users in the, mainly in Korea and US, and one institute in Europe. We have 2 installations at Eli Lilly right now, at Mayo Clinic, University of Michigan Ann Arbor, and UCLA right now, and 1 instrument in Belgium, as well as 4 more installations in Korea. So thank you for taking time to listen to our technology. Thanks. Oh, what booth for? 705, I think.